Okay, good morning, ladies and gentlemen. Let's start. We are starting our antibiotic resistance module this week. And I know you are frozen, your brain cells are frozen, so we start out with something that you need to think about. How many of you would like to be doctors here? How many of you are pre-meds? Oh, fantastic. Excellent. So you can actually practice that today. So imagine that you are a doctor and you prescribe an antibiotic for a bacterial infection. And I will show you a few graphs, figures, and you will need to draw conclusions based on them. So look at this one. This is patient 1. You see the number of cells growing in the blood. These are the days starting out right here when you collect the blood samples and you count the number of bacterial colonies. Think for like 30 seconds, what can you read from this figure? What do you think happened to this patient? All right, any ideas? Recommendations? Yes. Okay, so you're talking about day 12? Okay, so day 9, something. Thank you. So something definitely happened in day 9 when the bacteria was affected by the antibiotic. But that still doesn't explain what was going on. Oh, you said that it's exponential growth. Okay, so that could be an explanation. Any other scenarios? Anybody else came up with something different? Yes, sir. The patient is not taking the antibiotic. Okay, so he's saying that the patient is not taking the antibiotic day 6, 7, 8, and then feels better again, and what do you think happens here? Start taking it again. Excellent. Thank you. So these were the two scenarios I came up with also. So the first one, the first antibiotic was taken until day 5 and restarted again on day 9, so it's the same antibiotic maybe. Or there may be 2 different antibiotics. That's more related to what you were saying, is that it was taken until a point selected for resistant cells. It didn't really work, so the doctor went and prescribed another one that started to take at day 9 and hopefully did not finish just in a few days, but was taken for a longer time period. Right, so there are many different explanations thinking about antibiotic resistance and what happens to these bacteria. All right, so read this information, and first just please pick which one of these patients do you think, A or B, was affected by antibiotic resistance. All right, pick one, A or B. So based on that figure, and unfortunately I cannot put the figure up to the question, which one was showing resistance? right, 10 more seconds. All right, the majority of you said patient A. All right, this is the second question, and again, I'm going to get back to these answers later. I'm just curious whether what you think now and what you may think later. So if you can prescribe a new antibiotic to the patient with resistance, the one that you picked, which one would you prescribe, streptomycin or ampicillin? I give you a minute or so for this because there is information on this slide that you need to make this decision. You may actually need to look some information up online. All right, so hopefully you made a decision or an educated guess. And again, I'm just curious about your opinion here. All right, 10 more seconds. Oh, almost half and half. Okay, let's take like 30 seconds to explain to the person next to you or behind you why you picked that patient and why you picked that antibiotic. And if it was just an educated guess, just say that. All right, 30 seconds, go. All right, excellent. So hopefully you came up at least with an idea of what information you were supposed to be looking at on this slide, and we are going to get there in a moment to discuss that. So, in this module, we are going to cover certain things in lecture and also in lab before you jump in and design your own experiment. We are going to talk about mutations and bringing in some basics of genetics, talk about DNA replication, the central dogma today. Next week, visit Darwin and his travels, talk a little bit about evolution and selection types. Again, just touching it enough that you understand what you are doing in the lab sections. And then today and next week, we are going to discuss antibiotic resistance, talk about bacteria, how bacteria grows, and what kind of antibiotics you are going to use in lab. The learning objectives, or the big picture for this module, is really understanding how mutations influence the genotype and through that the phenotype. If we give you data, information, either on figures or in data sets that you collect, you will be able to make decisions whether you are dealing with spontaneous or induced mutations and knowing what the differences are. Understanding natural selection, and again, you are going to design and conduct an experiment in antibiotic resistance. And you will use your good old friends, the micropipetters. You're going to conduct serial dilutions, sterile technique, and you will be learning how to plate bacteria as early as this week in lab. So there were many pre-lecture questions, and I just wondered if you answered them correctly, and the only way we can see that, I take a look at them and discuss them very briefly. All right. Everybody answered this one correctly, so that's fantastic. Okay, so this was again a simple question. You could just look it up. You are going to have 4 different markings on these plates starting this week. So make sure that you remember which one means which antibiotic. All right, I will show you this in a figure in a moment, but we were missing only one phase. That was the lag phase. Again, this was a figure in the lab manual. The majority of you also answered this correctly, comparing aminoglycosides and beta-lactams. These are the 2 groups of antibiotics we are going to use in lab this week and next week, and you will see the differences, and you will have hands-on experience with them. All right, this is a little bit trickier. There are 12 different experiments you can actually conduct in this lab module, and they are in 5 different categories. And those categories are described in details in the lab manual. And make sure that you know which methods, either disk diffusion or colony count, you can use for those. So in this case, disk diffusion was the correct answer. Evolution in Action paper. Frankly, if you read that paper, you will understand everything about antibiotic resistance. It's a very well-written paper. It was posted in the additional readings in the ebook. And it describes what the challenges are with antibiotic resistance. So if you answered this question incorrectly, or you did not read the paper, you just guessed, please go back and read it. It's really useful, especially if you are pre-med or thinking about going to medical school. All right, this one was an interesting one. This is a direct question from the ebook. And if you went through this, it compared streptomycin and penicillin. And you have to be really careful. Many of you picked this, but we are talking about eukaryotic cells, right? Bacteria are prokaryotic. We have eukaryotic cells. So streptomycin cannot affect the protein formation in eukaryotic cells, otherwise it would kill us. That's why antibiotics only destroy bacteria and not the human cells. All right, excellent. So these are very rare mutations. Mutations are rare events. 1 in 10 billion nucleotides may be affected by the errors in the DNA replication. And again, this is just a comparison of gram-negative and gram-positive bacteria, and most of you got this correctly, and we'll discuss them in a moment as well. All right, when you design your experiments and when you come up with an experimental design and hypothesis, in the lab manual in Appendix 5, you will need to remove one of these pages and make sure you remove the correct one and turn it into your TA so you can request materials for next week. So it's very important that you will be able to pair up your experiment with the correct page. Otherwise, you will not request the correct materials. Okay. And pre-lecture questions for next week were just posted last night. So the readings for next week much shorter than they were for this week. You can probably finish them within half an hour. And there are some very simple questions. And these questions are not there to annoy you. And we are actually really nice people here. But they are to drive you towards the right direction about the readings. They are sort of just helping you go through the readings, look at material, and this way I can direct you what to look at, what the important material is for next week. So back to bacteria. This one was one of the questions about the lag phase, and lag phase is right when bacteria grows, starts to create enzymes, but it's not going through the exponential growth yet. Exponential growth happens right after the lag phase, and this is exactly where you are going to study your bacteria. And with Serratia marcescens, the bacteria we are going to use in lab, it goes up to a certain density by the stationary phase. And it's important because you are going to go through some basic calculations, and some of this information will be important to know. So again, lag phase, enzymes are being produced. Exponential phase, really that's when the bacteria is growing and reaches a stationary phase, and just like everything else on the planet, eventually dies. So exponential growth is really the phase when we are looking at this bacteria and you are going to study this bacteria. And just bringing in some calculations, you will be able to figure out how many generations bacteria went through, how much it grew, how many times it divided. And that's an important information when you try to figure out how many cells are on the plate and how many cells you pipetted onto the plate. So read this question. Think about it. Feel free to discuss it with your neighbor. I'll give you a minute or two for a quick calculation using that information that's on the slide, and there will be a poll question about it. So again, come up with a number. Check with your neighbor whether your neighbor came up with the same number. So again, the 22 hours is the total time from the moment that bacteria was born, going through the lag phase and exponential growth phase. How many of you are still calculating? Okay, I'll give you 1 more minute. Yes. You do not need to know the initial population. You're asking about the number of generations. It's not population density, it's about the number of generations. Right, this may be easier to say. Hopefully one of your numbers are close to these. If not, an educated guess always helps, or check with your neighbor. All right, 10 more seconds. All right, most of you said 26. Let's see if that's the correct answer. So the first step is that you are going to remove that 2.5 hours, right? Because that's the lag phase. There is no exponential growth in that time. There is no doubling. There's no reproduction. And then, since every 45 minutes there's a new generation, just need to divide the time period 45 and figure it out, that you will have 26 new generations in 22 hours. So most of you did answer this correctly. This question directly came from the antibiotic resistance worksheet that's actually in your lab manual as well as on Blackboard. So check out those questions. They will help you prepare for the next practical exam, and they will also help you prepare for the antibiotic resistance lab sections. Okay, so they are growing pretty fast. So why aren't we not walking in this bacteria knee-high? And as you properly guessed, because of antibiotics, right? Antibiotic against the bacteria. So there are many, many different groups of antibiotics. In class, you are going to work with these two groups, and you can separate them based on their modes of action. One are aminoglycosides. They prevent protein formation. And they affect all the bacteria the same way because all bacteria make protein. The other group are the beta-lactams. They do not affect all bacteria the same way because they are messing with cell wall formation. And some bacteria, which rely on this thick peptidoglycan layer of the cell wall, will be affected. Those are the gram-positive bacteria. Gram-negatives, however, they have the bilipid layers, and they will not be affected as much. Again, this is discussed in your readings. And the bacteria you are going to work with is Serratia marcescens. It's a gram-negative bacteria. So this is information that's important for you to think about when you are designing your experiments. because you can actually make some predictions based on this information. And the first question I asked you today, actually that was the second, that was the information you needed. You needed to know whether this is a gram-negative or gram-positive bacteria, and needed to know which one is an aminoglycoside. No, you didn't need to memorize it, but that was information that you probably needed to look up. Okay, so we are not teaching you everything here, but we are teaching you how to critically think about these problems and how to try to solve them. And you will run into these problems in your lab sections when you are designing the experiment. So think through these experiments before you jump in and start to conduct them. All right, so we have aminoglycosides, and we have beta-lactams and all other different kind of antibiotics killing these bacteria. So why are not all bacteria extinct then? And you also know the answer for that, because we are talking about antibiotic resistance. In order to understand antibiotic resistance, we need to talk a little bit about genetics and mutations. And the first step is to think about DNA replication. And many of you probably already know about DNA replication. And if you read the book, you saw that there were 3 different ways scientists were thinking about DNA can replicate. One is a semi-conservative method. The other one is a conservative replication, and a dispersive replication. As you see that with the semi-conservative, one of these strands creates the template for one of the new DNA, and the other strand creates the template for the other DNA. With a conservative replication, it's pretty much replicated, and the new strand, the new DNA is completely new. While with the dispersive replication, it's sort of these parts are copied left and right. So which one is correct? And may not all of you be able to answer this question, and that's okay because this is not for grade. But Poll Everywhere allows us to click on the correct answer. So if you are texting in, you will not be answering this question, and this question will not affect your grade. Okay, because I removed this question. But I was curious whether it works for you guys who are answering on your computers or smartphones, so you can click on the correct image and decide which one you think is actually the way DNA really replicates. And in your ebook, there was a nice little animation that asks you to choose how DNA replicates. So if you already went through that, you should probably remember it. Again, this is not for grade, but most of you chose it correctly. As you see that 243 of you picked semi-conservative replication, and actually that is correct. So one of those strands becomes the template for one of the new DNA, and the other strand becomes the template for the other new DNA. So, all right, so how does it relate to, how does DNA replication relate to mutations? And mutations are nothing else but just these alterations in the nucleotide sequence and some errors that may happen during DNA replication. And those errors can happen because of just random events, or they can be affected by mutagens. Right? Can you come up with some mutagens? What would be what we call a mutagen? Yes? Okay, ethylene bromide, right? So you will actually be using that or something similar in your human microsatellite DNA lab. And yes, you will be really have to be very careful not to touch it because it can be mutagenic. What else? Yes, ma'am. Okay, radiation itself, right? Thank you. So radiation can also alter your DNA, and if it survives, then it becomes mutagenic. Anything else? Okay, we can think about some toxins. You read it in the news. I remember when I was a kid, they even said milk was mutagenic. It's clearly not. But people keep coming up with some ideas what may create mutations. And those mutations can be either little, just in one nucleotide, or affecting a larger fragment of the DNA and chromosome, so they can be chromosomal mutations. Why does it matter? Big deal, right? DNA mutated. Well, it matters because after DNA replication, there's transcription, creating an RNA. And from that, through translation, there will be a protein. And those proteins can be messenger molecules in your body, they can be enzymes, and they really are the expression of what happens in the DNA. Are these proteins always affected? No, but sometimes they are, and that's when we can see the expression of this mutation. So when we talk about genotype, is what happens in that DNA. And when we talk about phenotype, is what you actually see and being expressed. So when you look at those plates in a couple of weeks, those agar plates, and you see them, they are different colors maybe, the bacteria, or they are different sizes. That's a phenotypic expression of that resistance. Even just the fact that they survived, that's their phenotype. Because it means that they mutated and they had antibiotic resistance. Right, so I just put up Paul Newman because you see that the expression of blue eyes, right, that somewhere is hiding in its genotype, but what you actually see are the blue eyes. It's his phenotype. But not every single mutation can be seen, right? So sometimes you have silent mutations. When there is no change in this polypeptide, there is no change in the amino acid sequence, there is no change in the protein. Mutation happens, you don't see it. We cannot see each other's DNA, we can only see the expression. So if we cannot see it, we don't know it happened. There are missense mutations, and there's actually one change here. And in most cases, this is what happens in antibiotic resistance. There are nonsense mutations when the mutation is not resulted in the production of a protein. So it really doesn't affect anything. They can be problematic if this is an enzyme, for example, that you need to produce and you need for survival. Then this can be problematic. But otherwise, there is no polypeptide that was mutated. And there's a frame shift mutation when we are not only talking about one single nucleotide, but by the time it gets to the mRNA and goes to translation, it affects a larger fragment of the DNA and therefore a larger fragment of your chromosome. And they can be really problematic in mutations. All right, this is really freaky, right? So these mutations, it's like we are surviving and what's going on. Well, how rare are mutations? Or how common are mutations? And you did look at the readings and you answered the question correctly. 1 in 10 billion nucleotides, maybe. There is some error in DNA polymerase, makes a mistake. And that may or may not turn into a mutation, right? So that was just an error. And there is maybe a polypeptide or a protein that shows. So they're really, really rare. That is exactly why you are not going to work with elephants this week in lab, but you are going to work with bacteria. Okay? Because bacteria do have really, really short generation times. And they are able to pass on that information in case there's a mutation with both bisexual and asexual reproduction. Right? So it was very nice. I wish it was closer to Valentine's Day, but this is the Valentine's Day for bacteria, and they use transformation, conjunction, transduction, and mostly binary fission to pass that genetic information on. So it's not only quick generation time, but also different ways to pass that information on. So even within a week, you are going to see mutated bacteria. With anything like us, with longer generation times, it's really much harder to study or even visualize what's going on. So in lab, it will be your time to study this phenomenon. Okay? So with this phenomenon is, hopefully you remember this, the process, the scientific process you need to follow, starting out from making observations, hypotheses, studying peer-reviewed journals, coming up, figuring out what has already been done, and conducting the experiment, and eventually actually producing either a paper or a poster at the end of the semester based on your study. So make sure you take this seriously. And again, I will say that a few times, but if you don't take anything else away from this course, I would like you to take that away, that you think and every experiment through. Don't just jump in and conduct an experiment. I've seen so many scientists failing because they did not really think about how many replicates they need. They didn't think about how are they going to analyze that data, what kind of statistical methods they may use. Don't make that mistake. Don't make that mistake in this class, and don't make that mistake in your scientific career. So make sure you think about everything before you jump in and conduct the experiments. And again, here it's low risk, right? So if you mess it up, no big deal. You won't have that beautiful data at the end. But if you work for a professor, or you are a professor, and you just conducted experiments for multiple years, and you realize you made a mistake with the experimental design, that can be really painful. And I've seen that happen. So the very first step is finding primary literature, and many of you have already done that. You are going to present 2 papers, or at least there will be 2 groups in every single lab, present 2 scientific papers, and on antibiotic resistance this week. And as I mentioned in a Blackboard announcement, you can either use just the library site and then go and search for papers, find the database, the Web of Science is really good for that, or go to Google Scholar. And a few years ago, Google Scholar was a big no-no, the evil. But now, actually, Mann Library and the Cornell Library System works together with Google Scholar to develop a new search engine. So it's easier for us scientists to find these primary literature. The challenge is that how are you going to start that? Right? How are you going to find these papers? And the good idea is to find papers by knowing exactly what you are looking for, coming up with these, with the search words or the proper keywords that you can search for. So since we have enough time, for those of you who have laptops or even smartphones, I challenge you, who's the first person who can actually find and download this paper? Okay? Go for it. I'm just curious how quickly you can actually find and download this paper. You know which journal it is in. You see keywords there. And again, if you don't want to compete with this, it's completely fine. There's no prize. You got it? Did you download it in some sort of format, or you just found it? Okay, so make sure you go to the next step. I want you to download it. Hands up if you are the first person who downloaded it. All right. What format do you have it in? PDF. How did you find it? You just Google the whole title. Okay. It works, but doesn't always work. Clearly, it worked. Anybody else who didn't just Google the title? Yeah, so if you didn't Google the title, how did you find it? Okay, fantastic. So which keywords did you Google? Okay, but you just typed it into Google. You didn't go to a library. You Google Scholar. Okay. Sir, did you use Google Scholar, or you just searched for? Just Google. Okay. Miss, your hand was up also. How did you find it? Okay, so in Web of Science, you can type that in the whole title. Did any of you actually go to the tuberculosis scientific journal or look for the author? Miss, your hand was up. How did you find it? Okay, fantastic. So what keywords did you use? Excellent. Thank you. So as you see that, this demonstrates that there's no one way to find it. I do not encourage you just to Google a title. You are lucky because here, actually, the internet knows that you are on a campus. So we have access by using Cornell's wireless system or even the wired system. We have access to many of these journals. I ask you to complete a project and you go home for your Thanksgiving break. At home, you are not going to be able to just access any of these journals. You will probably need to log in. You need to go through the proper channels. So we have a worksheet associated with finding primary literature. So please go and use that if you haven't found primary literature yet, or if you just Google primary literature. It's really important that you figure out a better way to find it. And I actually, if you wanted to find this paper, I would either go to the library website or to Google Scholar and probably search for streptomycin and quinoline resistance and find that paper. Since you know the authors, it's also great to just go and search for the authors and then find their papers. And very often you find similar papers which may be in your interest. And then eventually you download them in PDF formats and you have hundreds of papers. And I just suffered with this last week. I was submitting a paper and I did not have them in, usually I use EndNote, and I just did not have those references as EndNote. And I submitted it to one journal, to chemical ecology, but there wasn't enough chemistry in the paper, so they wrote back and they said this is not the appropriate journal. So I submitted it to another journal, which required a complete different formatting for the references. And I think I had like 45 papers in the references. So I had to manually go through and change each one of them. It's probably the most boring thing I have done in the past 5 or 6 years. It's hilariously bad. So please don't make that mistake. And usually I have my references in EndNote, but for this research project, I didn't. Library provides you many different ones. RefWorks is probably the easiest to use, and it's on all of the computers. It's really worth it. As soon as you start to use references, download them, have them organized by RefWorks or EndNote, and they will format it for you when you publish your paper in a scientific journal. Even if you download those papers and you keep them in these citation management softwares, we ask you to use the CSE format, or other journals may ask you to use the APA format. It's one click, and your references are going to be formatted that way. So after you find papers, you are going to design your first experiment. This week, we are going to provide you some scientific papers. You will have the chance to search for your own. Those scientific papers are also on Blackboard, so you can take a look at them and use them for your research project. But after that, you will design this experiment. And you design experiments in 5 different categories, and you have like 12 different narrower categories, and within those, you can also make some changes. So you will either look at minimum inhibitory concentration, which is really looking at concentration and bacterial susceptibility to antibiotics. You are going to compare low pigmented or red Serratia marcescens strains. You can compare the resistant levels of different colonies, picking different phenotypes, and then see whether those phenotypes actually mean different resistance. You can look at cross-resistance. What does cross-resistance mean? Anyone? What does cross-resistance? Okay, it means that if a bacteria is resistant to one antibiotic, it may be resistant to another one. So you can look at whether your streptomycin-resistant bacteria are going to show resistance in ampicillin or kanamycin. You use two different methods. One is colony count. That's really simple. You plate the bacteria, you count the number of surviving colonies. The disc diffusion method, you dip the little paper disc into antibiotics and maybe different concentrations of antibiotics and place them into the center of the plate. Make sure there is bacteria on the plate. And then you can measure the diameter of that zone of inhibition, meaning that you can compare the susceptibility of the antibiotics or look at how different concentrations affect the susceptibility or the sensitivity of these antibiotics. When you compare two groups, you have been using some of these statistical tests, t-test and rank sum test. Again, how do you decide whether you are going to use t-test or rank sum test? What's the magic word? Shapiro test, thank you. So you will need to use the Shapiro test to see whether it's parametric or non-parametric. If you look at correlations, then you can use the Spearman rank correlation test. And within those correlations, you may see this. There is a threshold where there is no growth yet. Then you can measure the concentration, antibiotic concentration, and zone of inhibition correlation here, and then it may reach a saturation level. So when you are designing these experiments, I want you to think about at which point you are going to pick those different concentrations. You would like to figure out this area, especially the minimum inhibitory concentration. Again, if you are pre-med, you are interested in that, that's something as a doctor you will be figuring out, especially if you are doing research for real life for antibiotics. Again, don't forget the difference between alpha level and p-value. Now, it's not for the practical exam, it's for your own experiment and for your own data that you are going to collect. And as a very last question, I'm just curious, and you can freely use your mind. We talked about mutations, talked about using mutations in experiments. Can you think of any mutations, true or not, in bacteria or in humans, that you would find beneficial? All right, it's working. So as soon as you answer, thank you very much for your attention, and have fun in lab this week.